Hello, everyone. Thank you so much for joining us in our Needham Healthcare Conference. Today we have Aglia Biotherapeutics that engineer human enzyme that could treat both oncology and rare disease. Our CEO today is Anthony Quinn that will be presenting, and thank you for joining us.
Yeah, thank you very much. It's great to be here. I'm looking forward this morning to telling you about the progress we've made at Aglia and also to share some recent data that was presented at the SIMD meeting on Sunday of this week on our lead program, pegzolarginase in arginase 1 deficiency. I will be making some forward-looking statements with risks and uncertainties, and I would refer you to our regulatory filings. Aglia is a clinical stage biotechnology company that engineers next-generation enzymes, human enzymes, with either improved properties or novel enzyme activity that provide solutions for diseases with high unmet medical need. We're based in Texas. We have, as of December 31st, we had $139 million in pro forma cash on our balance sheet. We're well poised for future growth. Our lead program, pegzolarginase, is currently entering phase 3 studies. We have 2 pipeline programs for more common rare diseases, homocystinuria and cystinuria, that are now in IND-enabling studies. And we have other research opportunities from our human enzyme-based DNA, our enzyme platform. We've had a lot of great momentum over the last 18 months. I'll share with you some recent milestones that are important. We over-enrolled our phase 1/2 study for pegzolarginase and arginase 1 deficiency. That data informed the design of our pivotal study, which we finalized at the end of last year. The data that came out on Sunday from our phase 1/2 study is very compelling and actually exceeded our expectations and actually gives us high confidence in our pivotal trial design. We presented some very compelling preclinical data for our cystinuria and homocystinuria at the end of last year. Those molecules have now moved into IND-enabling studies. In terms of upcoming milestones, we expect to dose the first patient in our pivotal study this quarter, and we will provide an update of our phase 1/2 clinical study with more repeat dose data at 20 weeks in the fourth quarter. We expect to file an IND or CTA for our homocystinuria program, and we will be giving an update on PK/PD, dose response, and bioavailability later in the year. Driving these accomplishments is a talented management team with deep and relevant experience across the drug discovery and development continuum. So before going into our new phase 1/2 data, I want to just remind you about 2 things. Firstly, why pegzolarginase is important for patients with arginase 1 deficiency. And secondly, I'm going to give you an outline of our pivotal study so that you can understand why our new phase 1/2 data is very important for our pivotal study. Arginase 1 deficiency is a devastating rare disease which first presents in children with serious manifestations. We estimated initially that there would be at least 600 patients in the major addressable markets. We've already found 170 patients, and based on some information that we've generated over the last 12 months, we now believe that population is at least 1,000 in the major addressable markets. The challenge for patients with arginase deficiency is that current disease management does not address lower plasma arginine levels, which is the key driver of the disease. And what that means is these children progress to very severe complications and with increased early mortality. Now, in order to understand our phase 1/2 data, I just want to spend a few minutes on our pivotal trial design, which we finalized at the end of last year. This is a double-blind, placebo-controlled study. We're recruiting 30 patients, 20 in active, 10 in placebo, and the double-blind period is for 24 weeks. The primary endpoint is plasma arginine reduction. The secondary endpoint includes measures of clinical response, including assessments of adaptive behavior and assessments of mobility, and you can see why those assessments are important in the data I'm just going to show you. So our new phase 1/2 data we presented in summary shows three things that are really important and underpin the design of our pivotal and give us great confidence in our pivotal design. Firstly, we can capture the disease burden in these patients in a very effective way using measures of mobility and adaptive behavior. Secondly, we have demonstrated that pegsilarginase has a transformative effect on plasma arginine levels relative to standard current treatment. And thirdly, we are seeing a very high clinical response rate in the phase 1/2 study in a relatively small number of patients dosed for 20 weeks duration. So in terms of baseline assessments, you can see here the mobility assessments include a timed walk test and gross motor function, which assesses other aspects of motor function. Just as an example, it assesses your ability to walk carrying an object when you're a bit distracted. Can you walk up and down stairs holding onto a handrail? Are you able to kick a ball with your left foot and your right foot? So it provides additional information on mobility relative to the 6-minute walk test. Using those combination of mobility assessments, 14 of the 15 patients in our phase 1/2 study had a deficit in mobility. We were able also to do assessments of adaptive behavior in 10 of the 14 patients at baseline. And for people who don't know what adaptive behavior is learned skills that are important in everyday life. They're grouped into 3 categories, practical, social, and conceptual. So let me just explain as a couple of examples. So as a couple of examples, the ability for somebody to follow a command, the ability of somebody when they're given something to say thank you, the ability to clean your teeth, the ability to dress, the ability to tie your laces. These are all examples of adaptive behavior which are assessed by questionnaire, and they were abnormal at baseline in 80% of the patients. Let's now look at the impact of pegsilarginase on arginine control. We now have 14 patients who have received 8 weeks of pegsilarginase and 10 patients where we have arginine data in the open label extension. At baseline, as I said earlier, these patients run very high levels of plasma arginine with a mean of 339. The current goal of therapy is to lower levels less than 200, and you can see none of the patients actually have levels well controlled in that range. We see decreases after 1 dose, decreases at 8 doses, which are both statistically significant, and after the 8th dose, the mean arginine level is 126 micromolar, which is just above the normal range. In the open label extension, you can see that with that longer term dosing, we see further improvements with a mean arginine level of 99 micromolars, which is within the normal range. So, pegsilarginase has a transformative ability to take arginine levels down from very high levels into the normal range in these patients, which cannot be achieved with current therapies. So now let's look at the impact of those plasma arginine reductions on important aspects of the disease. So what we presented on Sunday is we are now seeing substantial clinical responses in these patients accompanying these reductions in plasma arginine level. The graph on the right shows the mobility assessments. So we can see improvements in the mobility assessments after only 8 weeks of repeat dosing. With further dosing, we see further improvements, both in terms of non-responders at 8 weeks who then become a responder, or people who have responded at 8 weeks and show an increase in the magnitude of response with a longer duration of plasma arginine control. Looking at this in summary, in the 5 patients who have received 20 weeks of dosing, we had a 100% clinical response rate based on the combination of mobility assessments, timed walk test, GMFM, capturing complementary aspects of mobility. So the overall response at 20 weeks was 100%. In terms of the overall response at 8 weeks, what we see there is that if you just looked at mobility assessments alone, we have a 36% response rate, which is a good response rate given it's only 8 weeks. By including adaptive behavior, which is a different aspect of the disease, you can see that the response rate goes up to 57%. So what we can say is we're seeing a substantial clinical effect with pegsilarginase. We are seeing substantial effects on mobility and adaptive behaviour, which are able to capture that benefit using two very different aspects of this disease that affects children and causes devastating complications. The safety profile is great for moving into our pivotal study and moving towards registration. We've got really extensive clinical experience now with pegsilarginase. We've given more than 300 infusions. We've only seen 7 infusion reactions in 4 patients with pegsilarginase, and we're obviously very excited about moving this forward into our pivotal study. For success in a pivotal study in a rare disease, there's 2 things that you need. You have to have a good protocol, and we know we have a good protocol, and we know that our confidence in that protocol is reaffirmed by what we've seen with our phase 1/2 data that we presented recently. The second thing is having a good pool of patients where you're planning to do your clinical trials. We've had great momentum increasing our patient numbers over the last 18 months, and we've actually already identified more than 170 patients in the major addressable markets. These patients are mainly located in the US and Europe, so we're very confident that with that pool of patients and our protocol and the need for 30 patients, we're confident in terms of the recruitment of our pivotal study. So, where we are at the moment is we have a trial design. The phase 1/2 data reaffirms our confidence in the trial design with plasma arginine reduction as the primary endpoint, and secondary endpoints that include clinical responses assessed by adaptive behaviour and mobility. We expect to dose the first patient in the pivotal study this quarter. So let's now move on to our pipeline programs and provide a little bit more colour on our pipeline programs, which again are second generation enzymes, and in this case where we've actually been able to create novel enzyme activity that is actually a solution to diseases with high unmet medical need. So the first disease is cystinuria. This is a kidney stone disease. These patients develop multiple recurrent kidney stones from an early age. This leads to multiple episodes of severe abdominal pain, multiple hospital admissions, interventions and surgical procedures, and increases the risk of chronic renal failure and hypertension. We came up with a very elegant and simple solution which involves breaking down cystine in the plasma. Let me explain. In normal people, cystine is filtered into the urine. It's reabsorbed from the urine, so the levels of cystine in the urine are low. In patients with cystinuria, where there's a genetic transporter defect, they are unable to reabsorb the cystine from the urine. This leads to high cystine concentrations in the urine, and they get stones. So our elegant and simple solution was to lower plasma cystine levels, and by lowering plasma cystine levels, we should lower levels in the urine. That should inhibit crystallization and stone formation. So this is what we do with our second generation enzyme approach. So we take an enzyme, we basically reduce the activity of its natural substrate, and we're able to increase the activity against cystine. We then take that lead, improve the enzyme, and then we took that improved enzyme, optimized enzyme, into an animal preclinical study, and you can see very nice reduction of plasma cystine levels. So we next took this enzyme into a mouse model of cystinuria. These mice get stones, they get crystals in their urine, and they get kidney damage because of the stones. So you can see, as expected, this novel human enzyme reduces plasma cystine levels, reduces urine cystine levels, and the crystals that were present in the urine in the mice before dosing disappeared within 24 hours of the dose. So to finish off our profiling, we then took our enzyme into the mouse model and we dosed the mice for 6 weeks to demonstrate an effect on stone formation. So what you can see is these mice normally have very high numbers of kidney stones and high volumes of stones. And you can see with 6 weeks of treatment with our enzyme, we're able to lower the stone formation in these animals. So what we have is, we have a very innovative approach for patients with cystinuria. We've demonstrated efficacy in a preclinical model, and that model is highly translatable to the human disease, And as I mentioned earlier, we've actually moved that molecule into IND-enabling studies as we progress it towards the clinic. So let's move on to our next preclinical program, which also is a next-generation enzyme, and again has developed a novel enzyme activity from a human scaffold. So homocystinuria is another metabolic disease which has high unmet medical need with devastating complications. Again, it's a more common rare disease with more than 5,000 patients in the major addressable markets. And our goal was to develop an enzyme that was able to break down in the plasma homocysteine and its related dimer homocysteine. And the rationale was if we only had an enzyme that broke down homocysteine, the dimer would rapidly reform the monomer and keep the levels high. So we believe the best way to drive down homocysteine levels in a sustainable basis is to have a novel enzyme that breaks down homocysteine and homocysteine. So that's exactly what we've done. This enzyme we've confirmed is it lowers homocysteine levels in 2 different mouse models of homocystinuria. And we've also looked at the efficacy in the model which is shown here. So these mice develop an abnormal liver which rapidly deteriorates and the animals die as a result of the liver abnormalities and other complications. Treatment with our enzyme improves the liver appearance, actually resolves the abnormalities. In actual fact, the liver with long-term dosing, the histology is entirely normal. The improvement in the disease-related abnormalities is accompanied by a marked improvement in survival, so untreated mice die very rapidly, treated mice survive. So what we have is a novel human enzyme. It lowers plasma homocysteine and homocysteine levels. It corrects disease-related abnormalities in the disease and improves survival. Again, this model is highly translatable to humans, which gives us great confidence moving forward with this program. And we are in IND-enabling studies with this program, and we've guided to having our IND or CTA filed by the first quarter of 2020, and we'll provide an update on some of the pharmacology later this year. So in summary, where we are at the moment, we have had a really successful and great start to 2019, building on all the progress that we made in 2018, and we continue to deliver on the high potential of our human enzyme-based platform. With regard to our pivotal study, we have presented recently very compelling data that exceeded our expectations in terms of both the arginine lowering effects and the clinical responses, which gives us high confidence in the design of our pivotal study, which we'll be starting dosing on this quarter. Secondly, the experience in the organizational development that we've had as we've advanced pegsilarginase sets us up really nicely for taking those preclinical programs and advancing them through pre-IND enabling studies into the clinic. Thirdly, our lead molecule, pegsilarginase, and our preclinical programs, they nicely illustrate the power of our human enzyme-based research platform, where we're able to come up with solutions for diseases with high unmet medical need using an enzyme-based approach with either a better enzyme or an enzyme that has novel enzyme activity. And finally, we are very excited where we are at the moment, and we look forward to sharing data with outside as we move forward into 2019. Thank you very much for your attention.
What feedback did you get in terms of the way people were looking at the presentation from the other day?
I've been in biotech for a number of years, and one of the things that I always struggle with is the stock reaction to data. Sometimes we believe we have very compelling data. It exceeds our expectations. We are surprised with the stock reaction. I don't obviously have a clear understanding of exactly the dynamics of how that happened. Our job is to drive this program forward. We have a drug for patients with arginase 1 deficiency based on what we're seeing. seeing with the arginine reduction and these high frequency of clinical responses already. My job is to make sure we drive that forward through to registration so that we can actually get this drug approved for the benefit of patients.
What's the cash runway of the company? You probably said that. What's the cash situation?
So we have, at the end of December, we had 139 million in pro forma cash, and we have a cash runway Q1 of 2021.
Can you maybe tell us about the sales force preparation or thinking process and where to find these patients, for example, like regions that you'll be targeting first?
That's a very important question. So for arginase-1 deficiency in the US, we're actually in a much better situation than other rare diseases. There's always a problem with these very rare diseases. There's always misdiagnosis. There's always delays in the diagnosis. Where we are in the US is 34 of the 50 states are actually now doing newborn screening. Some of the thresholds are different between the states, so one of the things that we are trying to understand better is the potential for false negatives in some states relative to other states, but that's a really good place to be. There's also an ICD-10 code in the US specifically for arginase deficiency. Obviously you need to get the diagnosis to get that code, but it's a good starting point. So where we are at the moment is our initial focus was very much working with metabolic physicians. Obviously arginase deficiency is one of the urea cycle disorders. But the unique thing about arginase-1 deficiency is that it has very prominent neurological manifestations. And in actual fact, in some patients, they may not be aware that they're having they may not either be having hyperammonemic episodes, or they may not get diagnosed as hyperammonemic episodes. So the only thing you're aware of is that they actually have neurological complications. So our next wave of outreach is basically the pediatric neurology community, both in the US and Europe. We are aware of misdiagnosis of arginase deficiency. There was some cases have been misdiagnosed as cerebral palsy. We're also aware of a study from Brazil which was very striking in that 16 patients they looked at patients with end-stage neurological disease, and they basically asked the question, have we missed the diagnosis of patients with arginase-1 deficiency who have all been through the medical system? So in that hospital system, they found 16 previously undiagnosed cases of arginase-1 deficiency, which is why we need to get out there. So our goal at the moment with regard to that is MSL, medical affairs, MSL outreach, building a very small infrastructure that allows us to get the message out there to the metabolic physicians and to the pediatric neurologists. The great thing is having a drug that you are so very confident about where we are with that drug and our ability to get that drug approved. Obviously it gives us high confidence in terms of building that infrastructure so that we are in a really strong position for our commercial launch.
I saw that you have many partnerships with them. Can you tell us perhaps any partnerships in any of the clinical trials, and if they would be helping you with sales in other regions?
Yeah, I think the partnerships, those may be the logos of the companies on my team slide, of the companies that people worked at. Just to be clear, we own worldwide commercial rights for all of our programs, and we haven't partnered anything out at the moment. It's exclusive. We own all the commercial rights.
And would you be interested in a partnership like in the other regions besides the US, or is it in the talk, in the progress?
So with regard to rare diseases, clearly it's global development and it's global commercialization as you move forward. Obviously we understand the dynamics of our focus, and obviously initially is on the US and Europe. We understand those dynamics, and we'll be evaluating what's the best way to commercially launch this drug in the most efficient way possible, either doing it ourselves or actually doing it with somebody who is actually highly familiar like us with rare disease space but has a bigger infrastructure.
Can you also tell us about the cancer clinical trial that you have ongoing?
Yeah, so just the background for people for cancer, it's been known for a number of years that some cancer cells are very dependent on certain amino acids. There's two amino acids that kind of stand out. One is asparagine, and the other one was arginine. Obviously, there's a number of approved therapies to lower asparagine levels, different asparaginases, which are actually been either approved or actually been looked at in other indications at present. nobody's actually had a highly effective arginine metabolizing enzyme. And so basically what we've done at the moment is we've characterized that enzyme, we've demonstrated single agent activity in cutaneous and uveal melanoma. We have an ongoing clinical trial, clinical collaboration with Merck looking at combination with immuno-oncology approaches, because we had some very compelling preclinical data showing that we could actually enhance the efficiency of a number of different immuno-oncology approaches. So we're obviously running those trials at the moment, we'll be looking at the data, but our focus as an organization is to drive pegylaronidase through to approval in the rare disease indication. Obviously, it's eligible for a pediatric voucher. It's a quicker way to get to market, and we'll evaluate the potential upside with the pegylaronidase opportunity in oncology as we move forward.
Thank you, and that concludes our presentation. If you have any questions, you can ask outside of the Thank you.
Okay, thank you very much.
You got about 5 minutes? Yep. I'm going to use the study back here.